Good day, everyone. I am Rosita Mabolo from Microbiology major, and today I will be presenting my undergraduate thesis proposal entitled Assessment of Cytotoxic Potential and Antimicrobial Activity of Ehrlichia hiracifolius crude leaf extract. So here's the outline of my presentation. This would cover the study's background, objectives, methodology, and references. So for the background of the study, there are several problems that continue to plague the Philippine healthcare system. The cost of branded drugs in the Philippines is 22 times more than international reference prices, while generic drugs are 4 times more. This is according to Paris J 2019. Despite price reductions due to legislations such as Cheaper Medicines Act of 2008, as well as the Generics Act of 1988, those in the lower income bracket still cannot afford maintenance medicines for hypertension and diabetes, as well as antibiotics. According to World Health Organization 2019, traditional medicine is used by 60% of the world's population, and 80% of the population in poor nations rely almost totally on traditional medical practices, particularly herbal treatments, for their primary health care. Access to medicines and care from physicians and other healthcare professionals is especially challenging for geographically isolated and disadvantaged areas wherein the people are physically or economically inaccessible. Presently, plants are used in pharmaceutical industries for novel drug preparations to ensure efficacy and safety, as synthetic antibiotics are threatened for their multi-drug resistance. Here we have Ehrlichia hiracifolius as our plant sample. Ehrlichia is a genus of flowering plants in the daisy family known commonly as fireweeds or burnweeds. Ehrlichia hiracifolius belongs to the family Asteraceae. It is native to the Americas, but it is found in many places around the world, having been introduced by human activity. According to Morton J. 1981, Ehrlichia hiracifolius has been utilized in Venezuela and El Salvador for centuries. A plant used as a bath to alleviate fever and cough. Furthermore, according to Winlone 2021, burnweed has a number of medicinal properties. The oil of the plant has been used for treating wounds, poison ivy rashes, and some other ailments like piles. For additional information, as of this time, only mosquito larvicidal activity study exists. There is no existing study of cytotoxic potential and antimicrobial activity of Ehrlichia hiracifolius. So the cytotoxicity is an in vitro test to determine whether the plant extract will cause any cell death due to leaching of toxic substances or from direct contact. And on the other hand, antimicrobial assays are important tools to test and screen the inhibitory effects of compounds against microorganisms before establishing their inhibitory spectra. For the objectives of the study, this study generally aims to assess antimicrobial and cytotoxicity assay on crude leaf extract of Ehrlichia hiracifolius, and specifically, the study aims to first is to collect and verify the leaves of Ehrlichia hiracifolius, second to separate plant compound from plant material, and third is to assess antimicrobial activity and minimum inhibitory concentration of Ehrlichia hiracifolius crude leaf extract against E. coli and Staphylococcus aureus, and last to determine the cytotoxic activity of Ehrlichia hiracifolius through hemolytic activity. We are now down to methodology. So for our methods, here is the general experimental approach for my study. From collection and preparation of leaf extract to cytotoxicity assay, an antimicrobial assay will then follow. So first, we have the selection and collection of Ehrlichia hiracifolius leaves. So the samples of Ehrlichia hiracifolius will be collected from the public area of Barangay Dulugon, Maramag, Bukidnon. between the months of December 2021 to April 2022. The plant will be identified and confirmed by the University Museum at Central Mindanao University, Muswan, Maramag, Bukidnon. For sampling procedure, the young leaves of plants and other edible parts will be collected and washed thoroughly with chlorinated water. The samples will be spin dried and air dried. The dried samples will be set aside for different assays. So for the preparation of leaf extracts, with some modifications, extracts will be prepared using the method described by Pada and Picat 2008. In conical tubes, 0.5 grams of freeze-dried samples will be extracted with 12.5 ml of 100% methanol and 100% ethanol. The mixtures will be mixed for 20 seconds in a vertex mixer, then for 1 hour in a thermomixer at 850 rpm. The mixtures will be sonicated for 10 minutes and centrifuged for 10 minutes at 5000 rpm. The supernatants will be collected in separate conical tubes, and then the extracts will be kept at 4 degrees Celsius until they will analyze. For hemolytic assay, the cytotoxicity of Ehrlichia cerasifolius will be determined using a modified version of the method described by Rias et al. 2012, Pagano and Fagio 2013 with minor modification. The cytotoxicity will be studied by examining hemolytic activity against human red blood cells using Triton X 100 as positive control and 10% DMSO as negative control. The absorbance will be measured and then the cytotoxicity of the test samples will be determined by their percent of hemolysis. So for non-toxic, 9.44%, slightly toxic, 9.45% to 49.44%, and then toxic, 49.45% to 89.44%, and for highly toxic, 49.45% to 89.44%. And then the formula for calculating the percent of hemolysis is shown below. So for the antimicrobial assay, the antimicrobial activity of crude leaf extract will be tested on 2 microorganisms, the E. coli and the Staphylococcus aureus. So the dried extract will be dissolved in an aqueous solution of 50% dimethyl sulfoxide or DMSO in order to obtain the 5%, 10%, and 15% sample solutions. And as positive control, 2% of chlorhexidine digluconate will be prepared using the same vehicle. And then the DMSO solution will be used as a negative control. And then a volume of 10 microliter of each solution will place into individual sterile filter disc and allowed to dry at room temperature. And then each disc will place in the surface of Miller-Hinton or potato dextrose agar that had been previously inoculated with standardized inoculum suspension that matched the turbidity of the 0.5 McFarland standard or 1.5 times 108 CFU per ml by swab streaking. And then the diameter of zone of growth inhibition around each disc will be determined in millimeters after 24 and 48 hours of incubation. And then the test will be then performed in triplicate and 3 trials. A total of 2 isolates, one from gram-negative and one from gram-positive bacteria will be used in this study. So we are now on the data analysis. So this method is modified from Berman et al. 2018. So the data will be analyzed by using the MS Excel and presented as mean standard deviation of 3 replicates and 3 trials. So the one-way analysis of variance or ANOVA will be performed by using the StatPlus professional trial version software to determine the significant group differences, and means will considered as statistically significant if P is less than 0.05. And here's the references of this study. Thank you for listening and God bless us all.